The first step in protein synthesis is transcription. This begins at an initiation sequence, which is the TATA box. Transcription units bind, which allows RNA polymerase to attach and unwind the DNA strands. Next, in elongation, mRNA is formed through binds of complementary bases on the template strand. The sequence is terminated when a stop sequence is met. The mRNA dissociates. The next step is translation. A start codon of AUG begins translation, as a tRNA molecule carrying methionine bonds to the start codon of AUG. The job of tRNA is to carry specific amino acids to the codons so that they can form a correct polypeptide chain. Aminoacyl synthetase ensures correct binding between tRNA and its amino acid. A small ribosomal unit would have attached, and then a large ribosomal unit attaches. The first tRNA goes to the P site. A second tRNA bonds to the A site. tRNAs continue to bond to the A site, then move to the P site while adding its amino acid and a peptide bond and then exiting at the E site. As aforementioned, the aminoacyl synthetase joins amino acids to tRNA, and ATP provides the energy needed for this to happen. The polypeptide chain continues forming until termination, when a release factor binds to a stop codon. Hydrolysis occurs and the parts dissociate. Operons control gene expression. These are the components of an operon. Just upstream from the actual operon are regulatory genes. Regulatory genes code for repressors. Repressors, when they are active, bind to the operator and prevent transcription as they block RNA polymerase. The promoter, which is right here, is used in positive regulation as molecules can bind to it and increase transcription. Also, RNA polymerase binds to the promoter. The structural genes are what are produced during transcription. For example, in a lac operon, beta-galactosidase is the structural gene. One example of gene regulation is with the TRP operon. This is an example of repressible negative control. When the tryptophan is not present, the inactive repressor does not bind to the promoter, which allows additional tryptophan to be made through transcription and translation. RNA polymerase is not blocked. As you can see, the mRNA transcript translates into enzymes of the tryptophan pathway, which then makes tryptophan. When there is a sufficient amount of tryptophan, the molecule will act as a co-repressor, binding to the inactive repressor, causing a change in shape and making it active. This then will bind to the operator. This will prevent RNA polymerase from transcribing. Another type of gene regulation is with the lac operon. There are two different ways this can be shown, but the first is with a repressor, and it is an example of active repression. Initially, the repressor starts as an active repressor because the purpose of the operon in this case is to break lactose down into glucose by using beta-galactosidase, which is made during the transcription and translation process if RNA polymerase is able to bind to the promoter and complete this process. When there is no lactose present, there's no need to have beta-galactosidase because there's no lactose to break down into glucose. So when there is no lactose present, the active repressor binds to the operator. As you can see, the active repressor binds to the operator and RNA polymerase cannot move forward with transcription and translation. When glucose is absent and lactose is present, we do need the beta-galactosidase to break lactose down into glucose for energy. Lactose will bind to the active repressor, causing it to change shape and no longer bind to the operator. In this case, because it can no longer bind, RNA polymerase can go through, creating beta-galactosidase, which can then break the lactose down into glucose. When there is glucose but no lactose, the repressor stays bound and glucose is just used for energy. There's no need to make beta-galactosidase because there's no lactose. When there's low glucose, there's high cyclic AMP, which means that glucose, with its own source of energy, is losing energy. So when there is high cyclic AMP, it binds to the cap site. And the cap site, when the cyclic AMP is bound to it, is an activator, which means more beta-galactosidase is made, so that lactose can be broken down into glucose. With more glucose, there is less cyclic AMP, and there is no cap binding, so there is no production of the beta-galactosidase.